Hello, everybody. Thank you for the organization of this Congress, and thank you very much to giving us the opportunity to talk about biocatalysis. So today I would like to talk about sequencing platform, which is the Proteus by sequence methodology for biocatalytic process development from initial enzyme selection to ton scale production. So first I would like to talk about the group Sequence, which is a French company that is acting in pharmaceutical synthesis and specialty ingredients. We are more than 3,000 employees in Sequence. We have more than 24 manufacturing sites in Europe, North America, and Asia. And we have also 3 R&D centers, 2 in France and 1 in US. So the business of Sequence is roughly divided in 2 parts, half for pharmaceutical synthesis with a CDMO activity and some proprietary APIs, and also essential drug substances like paracetamol and aspirin, for which we are the world leader. The other part of the business is in specialty ingredients, where Sequence addresses different markets like healthcare, cosmetics, electronics, or food. And now I would like to talk about Proteus by Sequence, which is the R&D center in biotechnology for the group. So Proteus has been created 22 years ago. It has been acquired by PCAS in 2006. And then after acquisition by Novacap, we all became Sequence in 2017. So our job is to develop tailored enzyme and bio-based industrial solutions for the group Sequence, but also for external customers, since we are a CRO company. We are 25 employees at Proteus, multidisciplinary team with people skilled in chemistry and biocatalysis, fermentation, molecular biology, and also microbiology. And Proteus is located in Nîmes, south of France. So our activity is divided in 3 parts. The first is microbiology, where we exploit an exclusive collection of strains that includes extremophiles, microalgae, and among them, more than 1,000 have been fully sequenced. And so it gives access to an exclusive pool of potential of millions of enzymes. Another very important part of our work is the protein engineering for the tailor-made enzyme production. So for that, we use 2 proprietary technologies called Evosites and air shuffling. And my colleague will talk about that much deeper. And finally, the third part of our activity is the biocatalytic process development, where we implement our enzymes in chemical transformation. And since we are fully integrated into the Sequence group, we can transfer very easily our process to the manufacturing scale, meaning to the multi-ton production, thanks to the CDMO capabilities. So we have a library of enzymes, more than 350 enzymes that are ready to be screened. We have the important families, they are well represented. And over the past years, we have worked to select 7 enzyme families to propose them as kits. So the kits are called Sequenzyme, and we propose them to our customer for quick assessment of our enzyme on your target reaction. So concerning our capabilities, so at Proteus, we produce our enzymes at lab scale and up to 40-liter scales in bioreactors. And this scale allows us to have enough material for our process development, but also it helps to define a robust process to then transfer it to partners that can deal with industrial production of the enzyme at cubic meter scale. And in parallel, the enzyme is used at Proteus for the implementation of the biocatalytic process, and then we can easily transfer it to the Sequence lab at Porcheville, which is the Sequence R&D center in process development. And then the process can be upscaled and transferred to the pilot and manufacturers of the group for multi-ton production. So here is an example that depicts what I mentioned. It's the multi-ton production of D-serine, which is actually run at Sequence. So D-serine, the non-natural amino acid, is a building block for the synthesis of several APIs. It has also an efficacy for the treatment of tuberculosis and epilepsy. So the production of these amino acids relies on the condensation of glycine and formaldehyde, which is catalyzed by an aldolase. It's a very efficient process with a perfect enantiomer selectivity for the D amino acids. The productivity is very high since all the reagents are soluble in water and no protecting group are required due to the exquisite selectivity of the enzyme. This transformation was also exemplified to other aldehydes to give access to several unnatural D amino acids. And very recently, this transformation was also emphasized by an article paper in OPRD, where the D-serine produced by the process is the starting building block of an enzymatic cascade in which it's transformed into hydropyruvic acid, the substrate for a transcatolase, where carbon-carbon bond is created with several different aldehydes to produce 3S ketoses, and also non-natural and complex sugars. So this enzymatic cascade shows how powerful are the enzymes to create and promote complex transformations. Over the past month, and due to the COVID crisis, chemists at Sequence work also to review more than 100 of small molecules that are currently under repositioning for COVID-19 in order to identify alternative and more efficient synthetic routes. So danoprevir is one of these drugs where biocatalysis can help to get a more efficient transformation synthesis. So up to now, danoprevir is used as a protease inhibitor for hepatitis C treatment. And now there is a clinical trial ongoing on COVID-19 in China, where danoprevir is used in combination with ritonavir, another antiviral molecule. So danoprevir is a cyclic complex peptidic structure, which is made from 3 building blocks, which are amino acids, non-natural amino acids, and we focused on the building block number 2. So this amino acid could be efficiently produced by a rhodium-catalyzed asymmetric hydrogenation. It's an efficient process in terms of yield and selectivity, but there is a concern about the price of rhodium, which is constantly increased over the past years from a factor 8 to 13, depending on the years. So at Proteus, we proposed an alternative with the replacement of this precious transition metal by an N-reductase, which can catalyze the same reactions. And this time, an N-reductase is not affected by the sourcing of raw material like metal. And so we assessed our N-reductases on this transformation, and we saw that few of them were active on the reduction of these dihydroamino acids into the corresponding amino acids. So even if the conversion is not 100%, it's a very good starting point for optimization of the reaction conditions to go towards a very efficient process. So now I would like to talk about the Proteus by Sequence platform, which is used for biocatalytic processes development. So as I mentioned for danoprevir, the first step is to identify a first hit with an enzyme on the target reaction. For that, we can use sequencing kits for fast screening of enzyme on a target reaction, or we can also use enzymes that are described in the literature. And then it provides a first hit, and it's a good starting point for optimization of the reaction conditions. If the enzyme does not meet the requirements in terms of activity or selectivity, we can enlarge with diversity by giving access to our exclusive library of sequences. If it doesn't work, to find better candidates, we can also propose to optimize the enzyme with our directed evolution technology. Or alternatively, we can also propose semi-rational methods if the structural data are available for the enzymes. And so these 2 steps can lead to a more efficient candidates and can also generate IP on the reaction. And finally, with the selected enzyme or the optimized enzyme in hand, we can then develop the process of fermentation to produce the enzyme, and we can also develop the biocatalytic process to produce the chemical. And we generally use design of experiments in that steps to get a very robust process and go towards industrialization and get competitive process. As an example, for the first screening, we assessed the sequencing kit with the transaminase family for the amination reaction of this ketone into the primary amine. We get very good activity of our enzymes on the reaction for certain of the enzymes. And we also get, to our delight, perfect enantiomer selectivity for every active enzymes, giving the R or the S enantiomer depending on the enzymes. And so this quick assessment is a very good starting point for process intensification. And now I give the floor to my colleague to talk about biomolecular parts. Thank you very much.
So hello everybody, I will talk about the diversity of strains and enzymes at Proteus. We have a diverse collection of microorganisms coming from diverse partner and proprietary collections, which is a great potential for IP generation. This is done in respect with the Nagoya Protocol. More than 1,000 of these strains have been sequenced, which give us access to millions of exclusive sequences of enzyme. These enzyme and strains came from diverse environments in terms of temperature, salinity, and pH, as you can see on this slide. And this million of sequences can be exploited by bioinformatic tools for the selection and the screening of a desired activity. For a desired activity, we can enlarge the screening with a diversity of sequence, which is done by a bioinformatic platform tool named Genoscope. And this sequence came from, as I already said, from diverse microorganisms, including extremophile strains. The selection of enzyme sequences is done by a software named Cytoscape, which allows us to illustrate the selection and to select enzyme in terms of substrate specificity. And here we have an example of the transaminases available at Proteus, coming from 60 strains from the Proteus collection. Here we have an example of enzyme screening. Here, clients wanted to start directed evolution program for a target reaction. He had already several enzymes as a starting point. Here we have proposed to extend the screening to our own collection of sequence to identify a better starting point with maybe better activity and maybe a lower similarity with already patented or known sequences. A large number of sequences have been identified with putative desired activity, and we have selected 50 sequences representative of this diversity. These 50 enzymes has been compared with the already tested enzyme by the clients. We can see on this slide and the Cytoscape representation that the enzyme coming from the client selection very different in terms of identity from the whole selection of new enzyme. From the 50 new enzymes tested, we have identified 10 enzymes that had an activity on the target reaction, which was similar or higher to the best candidates coming from the client selection. And some of the best performing new enzymes have the weak amino acid sequence identity with the best performing enzyme from the clients. On this slide, we can see that the enzyme initially chosen by the client and has only 50% substrate conversion and is already known in patented sequences. And 3 of the sequences coming from the new selection have higher substrate conversion, and for one of them has only 50% identity with the patented sequence. So at the end of this screening, the customer and us chose an enzyme from the Proteus collection for a starting point for a directed evolution program. One of the directed evolution technology is named Evocide, and it consists of random mutagenesis. Here the starting point is only one gene. It consists of doing error-prone PCR condition determined using the Evocide algorithm in order to have from 2 to 10 mutations per genes. These PCR error-prone products are cloned in an appropriated vector, and the resulting libraries are sequenced to validate the mutation load. Once the mutation load is validated, each library is assayed with the appropriate activity test. We can then determine the active fraction of each library, which allow us, using the Evocide algorithm, to determine what we call optimal mutation load, which will be screened to find improved variants. Here we have an example of a directed evolution program using Evocide, which was made with Givaudan, and which is already published and presented previously. It consists of the directed evolution of a squalene open cyclase from Alicyclobacillus acidocaldarius for the ambrox production from homofarnesol substrates. We have performed one round of directed evolution, so with Evocide and random mutagenesis, and we have identified 3 variants with an improved activity by a factor of 1.6 to 3.5. And after an optimization designed by experiment of the reaction condition, we have obtained another 10-factor improvement for these variants. The second directed evolution technology is named H-shuffling, and it consists of random recombination. Here, the starting point is at least 2 genes that share at least 60% amino acid identity. These genes can come from public or Proteus sequence database if we have only one gene available. We can do sequence optimization to get higher nucleic acid identity to have a better recombination efficacy. And then the parental genes are cloned and the enzyme expressed to compare them to the genes to be evolved. Then we perform the parental gene random fragmentation using H-shuffling reaction. The resulting products are cloned in an appropriate vector, and the library are analyzed by sequencing to confirm the recombination events. And then the library are screened to find improved variants. Here we have an example of directed evolution program using H-shuffling technology, which was made with Firmenich. It consists of the production of green nodes. So as you can see on the left, you have the natural pathway for the production of green nodes in plants, and at the right of the slide, you have the reconstituted pathway. 3 enzymes are involved in this green node production: 13 lipoxygenase, which came from soybean flour in the reconstituted pathway, 13 hydroperoxidase coming from guava, which was a limiting enzyme and on which we have performed directed evolution, and a third enzyme, which is an alcohol dehydrogenase. So we have performed the directed evolution of the limiting enzyme, which was 13 hydroperoxidase from guava, which was expressed in Escherichia coli, and we have performed 4 rounds of directed evolution, mixing the hydroperoxidase from guava with 13 HPL from apple, orange, or tobacco, and 9 HPL from melon. During these 4 directed evolution rounds, we have screened 65,000 variants, and the best variant exhibits an activity improvement by a factor of 15. This best variant was also improved by a factor of 48 at 50 degrees. And this process is used at commercial scale by Firmenich.